Welcome to the InnoGuide podcast, where we share the works of authors and mentors to guide innovation. I'm your host, Bouthillier. This podcast, my website, and my newsletter are focused on helping you discover the secrets of innovation in the age of AI. Since I don't rely on paid ads, I want to thank customers who bring me onto their programs as a technical product accelerator, working alongside their teams to help them develop products smarter and faster without adding risk and while bringing human-centered AI skills into their organizations. You can learn more at innoguide.net. In this episode, number 106, we'll be sharing A World Built on Foundational Technologies by yours truly, Bob Bouthillier. This episode was inspired by Peter Attia's The Drive podcast, number 401, and his discussion of the foundational discoveries behind some of medicine's most important advances. What struck me was how rarely those discoveries begin with the product we recognize today. In one case, a scientist studied a fungus. Another studied snake venom. Someone else examined a bacterium living in a hot spring. But years later, those early observations led to statins, ACE inhibitors, PCR testing, and other tools that transform medicine. This made me realize that nearly every product we touch rests on work done by someone who couldn't have known where it would lead. The discovery may have looked narrow, strange, or even useless at the time, but its importance appeared later when another person saw a connection and asked a different question. Like, what else could this do? I suspect the AI tools we marvel at today may follow the same pattern. We see them as finished products because they can write, analyze, generate software, interpret images, and help us explore ideas. But they may be only the first visible layer of something much larger. A capability we treat as novelty today may become foundational to a product that hasn't yet been imagined. An unexpected model behavior, a limitation, or one useful insight buried inside a long conversation may become pivotal to a future developer. So this episode is about more than the history of medical innovation. It's about how discoveries travel, from nature into medicine, from physics and industry into medical devices, from signals we once ignored into tools that may help us understand health in entirely new ways. And finally, it's about what we must do now. Remain curious, preserve what we learn, and make it possible for the next person to find the insight we almost overlooked. Let's get started. After listening to Peter Attia's podcast, where he explored the foundational discoveries behind some of medicine's most important advances, it left me thinking about mold. Not the kind that forces you to throw out the bread, the kind that changes medicine. In the 1970s, Japanese biochemist Akira Endo began searching through fungi for compounds that might interfere with the way organisms produce cholesterol. His inspiration came partly from penicillin. If mold could produce one powerful chemical weapon, perhaps it had others waiting to be found. So Endo and his team began testing fungal cultures. Not 10, not 100, but thousands. After examining more than 6,000 samples, they isolated a promising compound from Penicillium citrinum, a fungus found in a rice sample obtained from a shop in Kyoto. The compound, later known as compactin or mevestatin, blocked an enzyme used in cholesterol production. It became the starting point for the statin medicines now used throughout the world. That is a remarkable journey. A fungus was protecting its own interests. Endo saw a cardiovascular drug. The medicine didn't begin with a neatly written product requirement. It began with the suspicion that nature might have already solved part of the problem. Sometimes, however, nature's solution comes in a less inviting package. Let's consider another case, the Brazilian pit viper. That's right, a snake. Its venom can cause a victim's blood pressure to collapse. That's obviously bad news if you have just been bitten. But to researchers studying blood pressure regulation, the venom revealed something valuable. Certain peptides in it interfered with the biological system that constricts blood vessels. Venom became a clue. And that clue eventually helped lead to captopril, the first widely used angiotensin-converting enzyme inhibitor, or ACE inhibitor. A substance designed by evolution to disable prey helped reveal a mechanism that could be controlled to treat hypertension and heart failure. The snake wasn't trying to become a pharmacist. Its biology simply exposed a lever that humans had not yet learned to pull. But there's another story from Atiya's podcast. There was a bacterium found in Yellowstone National Park. In the 1960s, researchers collected organisms from hot springs where the temperature was high enough to destroy most ordinary forms of life. One of those organisms was named Thermus aquaticus. At the time, it was an interesting bacterium living in a very strange place. But years later, became indispensable. The polymerase chain reaction, or PCR, works by repeatedly heating and cooling genetic material. The heating separates strands of DNA so they can be copied. Unfortunately, it also destroys ordinary copying enzymes. Early PCR, therefore, required a fresh enzyme to be added after each heating cycle. That was slow, awkward, and difficult to automate. But Thermus aquaticus had spent its evolutionary life solving the heat problem. Its DNA copying enzyme could survive temperatures that destroyed other enzymes. Once Taq polymerase was incorporated into PCR, the process could run repeatedly inside an automated thermal cycler. A bacterium collected because scientists were curious about life in hot water helped make possible modern genetic testing, infectious disease diagnostics, forensic identification, DNA sequencing workflows, and countless laboratory techniques. The hot spring wasn't the product, but the adaptation was. And yet there was another one on Atiya's podcast about the jellyfish. In 1962, Osamu Shimomura was studying how the jellyfish, Aquia victoria, produced light. He isolated a protein that glowed green. It became known as green fluorescent protein, or GFP. He received a Nobel Prize for this. For years, GFP was largely an answer to a question about jellyfish. Why do they glow? But the protein possessed an extraordinary property. The instructions needed to produce its green light were contained within the protein itself. That meant its gene could be attached to another gene and inserted into a living organism. The organism would then produce the target protein with a small green lantern attached. Researchers could then watch where it went. They could observe when a gene switched on, where a protein accumulated, how a cancer cell behaved, or how a neuron developed. A curiosity from a translucent jellyfish became a way to make invisible biological processes visible. That's the pattern Peter Atiya's discussion brought into focus for me. The original discovery rarely announces its future. The fungus doesn't arrive labeled cholesterol medicine. The venom doesn't say blood pressure pathway. The hot spring bacterium doesn't say global diagnostic platform. And the jellyfish doesn't say tool for watching genes inside cells. That meaning appears later. Someone working in another field sees more than the original observation. They recognize that the mechanism can travel, and that may be one of the defining characteristics of foundational technology. It doesn't solve only one problem, but it becomes a building block that other people can combine, adapt, and redirect. And most of the tools we touch today have stories like this buried underneath them. The medical device on a hospital cart may depend on a material developed for another industry. The sensor inside a wearable may descend from technology created for communications, photography, navigation, or manufacturing. And an algorithm may trace its lineage to researchers trying to recognize handwriting, translate languages, or identify objects in ordinary photographs. The polished product hides the archaeology. By the time we see it, the earlier discoveries have been integrated so completely that they appear inevitable. But they were not inevitable. Someone had to notice the fungus, preserve the bacterium, study the glow. And someone else had to look at that work and ask a different question. What else could this do? Could this solve my problem? This is also how I think about the artificial intelligence tools we're using today. We tend to describe them as the breakthrough, and they are remarkable. They can generate software, interpret images, analyze documents, create simulations, and help us explore ideas at a speed that would have been difficult to imagine just a few years ago. But I suspect they are not the destination. They may be the mold, the venom, the hot spring bacterium, or the glowing jellyfish of our time. Some capability we currently treat as novelty may become the foundation of an industry that doesn't yet exist. A limitation we complain about may force someone else to invent a new architecture. An unexpected model behavior may reveal a completely different way for humans and machines to work together. Today we may barely notice it. Years from now, a developer may recognize what it was really for. Some of medicine's most powerful tools did not merely emerge from nature. They turned nature's own machinery into something we could program. Chapter 2. Nature becomes a toolkit. In 1975, two scientists faced a problem that sounded deceptively simple. The human immune system can produce an extraordinary variety of antibodies. Each one is shaped to recognize a particular target. That makes antibodies wonderfully precise. It also made them difficult to manufacture. An ordinary antibody-producing cell could create the antibody researchers wanted, but it would not live forever in a lab dish. A cancer cell could divide almost indefinitely, but it did not produce the useful antibody. Georges Kohler and César Milstein wondered what would happen if they combined the two. The result was a hybrid cell, a hybridoma, that inherited the best and worst features of each parent. It produced one specific antibody, and it could keep dividing. For the first time, researchers had a repeatable way to manufacture large quantities of identical or monoclonal antibodies. At first, this was a laboratory technique. Then it became a platform. Monoclonal antibodies could be used as highly selective probes in diagnostic tests. They could help identify cells in blood and tissue. They could carry a signal that made a particular biological target visible. Eventually, they could also become treatments, attaching to specific receptors, blocking disease pathways, marking harmful cells for immune attack, or delivering a therapeutic payload. Today, entire categories of cancer, autoimmune, inflammatory, and infectious disease therapies depend on the idea that began with two cells that possessed complementary shortcomings. Neither cell was sufficient, but the combination changed medicine. A similar story began with something bacteria do when they're attacked by viruses. It took scientists years to understand what they were seeing. Researchers had noticed unusual repeating sequences inside bacterial DNA. Between those repetitions were short stretches of genetic material that appeared to come from viruses. The pattern was strange, but its purpose was unclear. Eventually, the mystery came into focus. Bacteria were keeping records. When a virus attacked, parts of its genetic code could be stored inside the bacterium. If the same virus returned, the cell could use that stored sequence to recognize the intruder and guide molecular machinery to cut its DNA. It was an immune system, just not ours. Emmanuel Charpentier and Jennifer Doudna helped reveal how one version of this system, CRISPR-Cas9, could be simplified and reprogrammed. Instead of directing the molecular scissors toward a virus, researchers could provide a different genetic address and tell them where to cut. They received a Nobel Prize for this. A defense mechanism used by microbes became a tool for editing the genomes of plants, animals, and human cells. That punchline is easy to state now, but it would have sounded absurd at the beginning. Scientists studying why bacteria survived viral attacks were not setting out to create a programmable editing system for inherited disease. The useful idea was hiding inside a biological feud that had been underway for billions of years. Nature had already built the mechanism. We learned how to change the destination. The story of messenger RNA is different because the basic concept was understood for decades, yet the practical problems seemed determined to keep it from becoming medicine. Messenger RNA carries temporary instructions copied from DNA. It tells the machinery inside a cell which protein to produce. That created an enticing possibility. Instead of manufacturing a complex protein in a factory and delivering the finished product, Perhaps we could deliver the instructions and let the patient's own cells make it. There were only a few problems. The RNA was fragile. It could be destroyed before reaching the cell. It had difficulty crossing the cell membrane. And the immune system could react to synthetic RNA as though it were a dangerous invader. In other words, it was an elegant idea with terrible shipping. Researchers kept working on both halves of the problem. Work on modified nucleotides showed that changing some of the RNA's molecular building blocks could reduce unwanted immune activation while improving protein production. Meanwhile, decades of research on lipids produced tiny particles capable of enclosing fragile genetic material, protecting it and helping it enter cells. The messenger finally had an envelope. When COVID-19 appeared, the vaccines seemed to arrive with astonishing speed, but the apparent overnight success rested on years of RNA biology, nucleoside chemistry, lipid research, manufacturing development, and earlier vaccine work. The emergency did not create the foundation. It revealed how much of the foundation had already been built. That's worth remembering because the most visible product often receives credit for everything beneath it. We see the vaccine, therapy, test, or device at the moment it becomes useful, but we rarely see the long series of discoveries that made it possible. And some foundational technologies do more than create one successful product. They change the way future products are built. A conventional medicine is usually designed around a particular therapeutic compound. A platform technology creates a reusable method. With monoclonal antibodies, the target can change while much of the production principle remains. With CRISPR, the genetic address can change while the underlying editing mechanism remains. With mRNA, the encoded instructions can change while much of the delivery platform remains. The reusable part is what allows the discovery to propagate. This idea appeared repeatedly in the original discussions behind this episode. Foundational discoveries gain enormous value when they stop being isolated findings and become building blocks that other people can direct toward new problems. The first useful application may therefore be the least important measure of a foundational technology. PCR became more than one genetic test. GFP became more than an explanation for glowing jellyfish. Monoclonal antibodies became more than a laboratory supply. CRISPR became more than a bacterial curiosity. Messenger RNA became more than a temporary message inside a cell. Each tool became a tool for making other tools. And medicine has another habit that may be even more surprising. When nature does not provide the entire toolkit, medicine borrows from somewhere else. The next chapter begins beneath the ocean, where sound was used to find objects that couldn't be seen. It then travels through factories, physics laboratories, space programs, and consumer electronics before returning to the examination room. Because some of medicine's most familiar instruments were not originally medical at all. Chapter 3. Medicine borrows from everywhere. In 1946, two groups of physicists discovered that atomic nuclei could be persuaded to behave a little like microscopic radio transmitters. Place certain atoms in a strong magnetic field, strike them with radio waves, and they respond. The response could reveal something about the atoms themselves. At first, this was not a medical imaging system. It was nuclear magnetic resonance, a way for physicists and chemists to study matter. It could help identify chemical structures. It could tell researchers something about the composition of liquids and solids. It could not show a physician what was happening inside a living patient. That required another leap. Paul Lauterbur realized that if the magnetic field varied across space, the returning signals could reveal where they came from. Peter Mansfield helped develop the mathematics and methods needed to make the process faster and more practical. A technique for studying atoms became a way to map the human body. By the early 1980s, MRI systems were entering clinical use. Today, they can distinguish soft tissues, reveal tumors, examine joints, map the brain, and show structures that would otherwise require surgery to investigate. The physicist had not set out to build a hospital scanner. Medicine borrowed their discovery. Ultrasound, though, followed a very different route. Its story begins beneath the water. When something is hidden in the ocean, light is not much help. Sound travels further. Send out a pulse, wait for an echo, and the return can reveal something is there. That principle became central to sonar. It was used to find submarines, map the seafloor, and measure distance underwater. But factories found another use for it. Sound waves could be sent through metal to reveal cracks, voids, and imperfections that could not be seen from the surface. A technology developed to find hidden objects became a way to find hidden flaws. Then medicine borrowed it. Instead of searching through water or steel, ultrasound systems sent high-frequency sound into the body. Different tissues reflected the sound differently. The returning echoes could be assembled into an image. The machine could show a fetus moving before birth. It could reveal the chambers of a beating heart. It could examine blood flow, guide a needle, or help a physician look beneath the skin without making a cut. The same broad idea had traveled from ocean warfare to industrial inspection to the examination room. But ultrasound has an unusual limitation. The machine could become portable, but the expertise couldn't. A skilled sonographer does not simply place a probe on the patient and wait for the picture to appear. The operator searches. The probe moves, rotates, tilts, and changes pressure. Anatomy may briefly appear, then disappear. Bone blocks the sound. Air scatters it. Body shape changes the path. The operator is part of the imaging system. But that's now beginning to change. AI-assisted ultrasound can analyze the image while it's being acquired. It may help determine whether the correct anatomy is visible, whether the view is good enough, or which direction the probe should move next. Semantic segmentation can outline anatomical structures. Software can propose measurements or identify that the image should be reacquired. The ultrasound machine is beginning to absorb small pieces of the expert's technique. That does not eliminate the expert, but it changes what the instrument can help another person accomplish. And then the ultrasound story took a very unusual turn. A company known for creating imagery pictures decided to enter medical imaging. Midjourney is that company. They became famous for generating images from written prompts. Ask for an astronaut in a medieval painting and the software invents one. It is skilled at creating images that never existed. But now Midjourney Medical is working on the opposite problem. It wants to reconstruct an image that must be true. The company has shown a prototype whole-body ultrasound scanner built around 40 ultrasound-on-chip modules supplied by Bouthillier Network. A person is positioned in the water while the system collects acoustic information from many directions. The prototype combines roughly 500,000 sensing elements with enormous processing capability. Then it attempts to reconstruct a three-dimensional view of the body. The idea sounded a little like medical sonar, except instead of one transducer moving across the body, many modules listen from around it. And instead of relying entirely on one operator to find each view, computation tries to assemble the echoes into a larger map. The project is ambitious and still experimental. A prototype is not proof that the system can replace MRI, CT, or conventional diagnostic ultrasound. Medical reconstruction has a stricter requirement than commercial image generation. A beautiful mistake is still a mistake. That may be the most interesting part of the story. Midjourney's consumer product is rewarded for imagination, but its medical work will be judged on fidelity. Can a company that became skilled at teaching computers how images look apply that experience to match the harder problem of determining what's really there? That answer isn't yet known. But the attempt is possible only because several earlier technologies have converged. Sonar taught us to locate hidden objects within echoes. Medical ultrasound taught us to form images from reflected sound. Semiconductor manufacturing allowed ultrasound transducers to move onto chips. And high-performance computing made large reconstructions practical. AI learned to identify and segment structures inside complex images. No single discovery created the scanner. The scanner sits at the intersection of all of them. This same pattern appears elsewhere, and now we'll talk about it with respect to the tiny cameras that are now used in medical products. In the 1990s, engineers at NASA's Jet Propulsion Laboratory advanced CMOS active pixel sensors because craft needed cameras that were smaller, lighter, and less power-hungry. Those sensors later helped make cell phone cameras almost unavoidable. They also found their way into medicine, though. NASA documented an early application in digital dental radiography where compact CMOS sensors could capture images inside the mouth. The same broad family of technology has helped make cameras small enough for endoscopes, surgical tools, inspection devices, and portable diagnostic equipment. A camera designed with space constraints in mind became useful in the most confined spaces of the human body. That is what medicine does particularly well. It borrows. It borrows from physics labs, naval research, factories, semiconductor companies, space programs, and consumer electronics. Then it places those technologies beside a clinical problem and asks a new question. Could this help us see what was previously hidden? And increasingly, the answer is not coming from a better lens, a stronger magnet, or a more sensitive probe alone. It's coming from computation. In the next chapter, we will look at signals that medicine once ignored because they seemed too weak, too noisy, or too ordinary to matter. The light scattered through tissue, the changes hidden in a person's voice, the daily fluctuations captured by a wearable. Signals that may have been telling us something all along, but we simply did not know how to listen. Chapter 4. The Signals We Used to Throw Away. Shine a flashlight through your hand. The light doesn't pass cleanly through. It spreads. It bends. It scatters in so many directions that the result looks like a reddish glow. For most imaging systems, scattering is a problem. A camera works best when light travels in a reasonably predictable path from the subject to the detector. Inside the body, though, tissue scrambles that path. The useful information appears to be lost. But what if it's not lost? What if it's merely difficult to reconstruct? That question sits behind some of the work at Open Water, a company exploring medical systems built from light, ultrasound, semiconductor technology, and computation. Its premise is that advances in manufacturing and processing may make it possible to manipulate and measure energy inside the body with devices that are dramatically smaller and less expensive than traditional hospital equipment. Near-infrared light can travel farther through tissue than ordinary visible light, but it still scatters. Blood flow and motion also change the returning optical signal in subtle ways. Those changes may look meaningless to the eye, but they can contain information about what happens beneath the surface. The camera alone can't solve the puzzle. The software must work backward. It receives the distorted pattern and asks what arrangement of tissue, blood flow, motion, and energy could have produced it. In some approaches, focused ultrasound can help identify a particular region within the tissue, while optical detectors listen for the tiny changes associated with that location. The limitation becomes part of the instrument. Scattered light, once treated as something that ruined the image, becomes a source of information. That is the kind of reversal that often precedes a foundational technology. Something we once tried to remove becomes the thing we learn to measure. Now, consider yet another signal we produce every day, our voice. Most of us think of the voice as a delivery system for words. It carries ideas from one person to another with the meaning in the sentence. But before a word leaves your mouth, several parts of the body have to cooperate. Air moves from the lungs. Vocal folds vibrate. Muscles shape the sound. The nervous system coordinates the timing. The brain selects words and organizes the thought. A change anywhere along that chain can leave a trace. Not necessarily one person can hear, but a computer may notice it. Researchers are examining vocal characteristics such as pitch, rhythm, pauses, articulation, breathiness, tremor, and speaking rate. Other systems look at language, word choice, and the organization of speech. The hope is that changes in these patterns may eventually help monitor neurological, respiratory, cardiovascular, cognitive, or behavioral conditions. The microphone is not new. The signal was always there. What is new is the ability to measure thousands of small features, compare them over time, and search for patterns too subtle for ordinary listening. But this field has a problem. Researchers do not always mean the same thing when they use the term voice biomarker. One team may be studying the vibration of the vocal folds. Another may be measuring pronunciation, while another examining pauses or vocabulary. Their work may sound comparable, even when they are measuring different biological processes. That is why the VOCAL, VOCAL, initiative may prove more foundational than any single disease detection model. VOCAL stands for Vocal Biomarker Guidelines for Ontology, Classification, Application, and Logistics. It's an international effort to create shared definitions and organize the field around clearer categories. Its first stage focused on establishing consensus terminology for vocal measures and biomarkers. That may not sound dramatic. There's no glowing scanner, no impressive robot, no announcement that a 30-second recording can diagnose everything before breakfast. Just a group of experts agreeing on what the words mean. And yet medicine cannot build dependable evidence if researchers are measuring different things using the same name. Sometimes the foundational discovery is a molecule, but sometimes it's a standard. The next signal is probably sitting on someone's wrist right now. A smartwatch or a ring may collect heart rate variability, movement, sleep, respiratory patterns, and sometimes even skin temperature. Each measurement seems useful, but each is frustratingly ambiguous. A higher resting heart rate may signal illness, or exercise, or stress, alcohol, or poor sleep. A lower heart rate variability may suggest that the body is under strain, or it may simply reflect an unusually difficult day. Skin temperature may rise with infection. It may also rise because the room is warm. One signal tells a weak story. Several signals changing together may tell a stronger one. Researchers studying early illness detection have therefore looked for combinations. Changes in resting heart rate, respiratory rate, temperature, sleep activity, and recovery relative to a person's own baseline. Studies have found that wearable data can reveal physiological changes associated with infection, while also showing that the signals are rarely specific enough to identify one illness by itself. That distinction is important. The wearable may not say, "You have this disease." It may say, "Something about you has changed." My mother could do that, but that could still be valuable. Traditional medicine often compares a patient with a population range. Continuous sensing creates another possibility, comparing the patient with themselves. Heart rate that appears normal on a chart may be unusual for that person. A temperature change too small to qualify as a fever may still be meaningful when it arrives besides your poor sleep, reduced activity, and a rising respiratory rate. The discovery is not one new sensor, it's the pattern between them. There have been some discussions about a future continuous diagnostic mesh in which light, sound, voice, wearables, imaging, and other health data become parts of a connected picture rather than an isolated environment. We're not fully there, but the direction is already visible. The National Institute of Health is developing its PriMed-AI program around a similar idea, combining clinical imaging with other health information to create more reliable and useful decision support tools. The image may be interpreted alongside laboratory data, clinical history, or other signals rather than treated as a separate island. This is where multimodal foundation models become interesting. A traditional imaging model might examine one scan and answer one narrow question. But a multimodal model may eventually connect the image with the patient's history, or prior studies, lab findings, wearable trends, or clinical notes. That sounds like progress, but it also creates a new danger. A model can be misled by the information around the image. Research has shown that multimodal systems may rely heavily on accompanying text, even allowing an incorrect suggestion in the text to weaken an otherwise correct image interpretation. More information doesn't always produce more truth. Sometimes it produces a more convincing mistake, though. That may be the central challenge of this new era. We're learning to recover meaning from signals we once ignored, discarded, or treated as noise. But once a machine finds a pattern, we still have to determine whether the pattern is real. Does it matter? And what, if anything, should someone do about it? The scattered light may reveal blood flow. The voice may reveal change. The wearable may reveal a deviation from baseline. The image may gain meaning from the information surrounding it. Each signal offers a clue, but the breakthrough may only come when someone discovers how the clues fit together. And the clue that matters most may be the one a person noticed unexpectedly, recorded carefully, and felt safe enough to share. Chapter 5. The Discoveries We Are Creating Right Now. In the last chapter, we looked at signals medicine once ignored. Scattered light, subtle changes in the voice, small shifts in heart rate, temperature, sleep, and activity. None seemed especially meaningful by itself. But then computation gave us a way to look at these again. The same thing is happening inside our organizations now. Every day, engineers, clinicians, designers, developers, product teams are generating observations at a pace we've never seen before. Some come from experiments, some from failed tests, some emerge during design reviews, and increasingly, many appear during conversations with generative AI. Person asks a question. The model offers a connection. That connection leads to another question. A possible design emerges. Then the window closes, and the insight disappears. That may be one of the greatest ironies of the AI era. We're creating more information than ever before, but we may be preserving less of what matters. The fungi, bacteria, venom, fluorescent proteins, sonar systems, and imaging technologies we discussed earlier became foundational because someone recorded what they found. The discovery survived long enough for another person, often in another field, to recognize what else it might do. Today, the next foundational insight may not be sitting in a scientific journal. It may be buried in an AI chat. A developer may discover that a model fails in a very particular way, but that failure may be useless for the current project, yet important to someone building a safer system later. An engineer may ask an AI assistant to compare two sensor technologies and uncover a relationship that was not part of the original assignment. A clinician may notice that patients describe the same symptom differently than the product team expected. A prototype may produce a strange result that turns out to be more interesting than the result the team was trying to obtain. The challenge is not simply to generate these insights, but it's to make them findable. A system such as Confluence, Notion, or another shared knowledge platform can help, but only when it holds more than finished reports. It should also preserve the learning that happens along the way. What did we expect? What actually happened? What surprised us? What failed? What question did it create? Where else might this be useful? Those small records may look unimportant when they are created, but so did a glowing jellyfish. But a knowledge platform is not enough. People also have to feel safe using it honestly. I once worked with an organization where the public signs of low psychological safety were difficult to ignore. It had a Glassdoor rating below 2.5, and most employees reviewing the company did not recommend working there. It was too bad. Although that matched what I saw inside. Innovation still happened, but it happened below the radar. People experimented quietly within their own groups. They shared ideas with people they trusted, but very little traveled beyond those boundaries. In one case, an AI tool costing about $30 a month could have substantially improved an HR function serving more than 500 employees. But no one requested it. $30 wasn't the obstacle. The risk of asking was. That's what low psychological safety does. It changes the question from, could this improve the organization, to, what happens to me if this attracts the wrong attention? The company didn't reject the experiment. The experiment never became visible enough to be rejected. And that's how organizations lose discoveries. Not always through a bad decision, but sometimes through silence. This becomes even more important as AI moves beyond the prompt window. Today, many of us experience AI as a tool we visit. We open a chat, ask a question, review the answer, and close it. But agentic systems, they'll be different. They will surround our work. They may read documents, monitor projects, generate code, analyze data, call other tools, results, prepare reports, and move tasks from one stage to the next. They will behave less like one assistant waiting for a question and more like a group of junior assistants working continuously around us. That offers enormous leverage. These assistants may have access to libraries of information no person could ever read or remember. They may connect an old finding to a new problem. They may surface a forgotten experiment. They may help us see relationships we would otherwise miss. But access to a library is not the same as judgment. A junior assistant can research, compare, draft, and recommend. But it should not silently decide which outcome the organization accepts. We will need to know which AI tools are being used, what information they can access, which tasks they're allowed to perform, what work requires human review, and which outputs become part of the controlled development record. This is where the idea of an AI control tower becomes useful. A good control tower should not automatically send every flight to the airport with the most runways available. It should know the actual destination and route it appropriately. Some destinations require a large international airport. Others are better served by a regional field. In some trips, they should never involve an aircraft at all. We're going across the street for a sandwich. AI should work the same way. Some tasks require a frontier model. Others can be handled by a smaller, faster model. Some are better served by deterministic software, an approved workflow, cached information, or a result that already exists in the company's knowledge base. The objective is not to use the cheapest option every time. It's to use the least expensive and the least resource-intensive approach that can reliably produce the required outcome. That matters to the business. An AI-enabled education product may deliver real value, but it can't grow sustainably if every interaction is sent through an unnecessarily expensive model. The technology may work perfectly and still fail as a product because the economics don't scale. This also matters, though, environmentally. Every unnecessary model call consumes computing resources and energy. The exact impact depends on the model, the hardware, the provider, the workload, and the energy source. But the operating principle is simple. Don't use more computation than the outcome requires. Cost, latency, energy, and environmental impact are not separate conversations. They're all influenced by the routing decision. This is what led me to create GetWrangler.ai, because I couldn't find another solution that was both fast enough and comprehensive enough for my client applications. Routing important work to a model that's not capable enough gives you poor outcomes. It results in rework, dissatisfied users, and if the outcomes aren't caught before they go out the door, it can cause brand debt. Routing ordinary work to a model that is far more capable than necessary keeps costs and resource use unnecessarily high. Neither is good architecture. A control tower should help the organization reach the correct destination, preserve a record of how it got there, and do so with no more cost or energy than the journey requires. That record may become particularly important in medical product development. AI is already helping teams research technologies, draft requirements, analyze risks, generate software, create test cases, investigate failures, and prepare documentation. Even when a human reviews and approves the final work, the model may have influenced how it was produced. In the future, it may be important for medical products design and development history to show which models assisted with different parts of the work, who used them, and which project they supported. That does not mean every prompt belongs in a design history file, but it means the organization should be able to preserve provenance when AI materially contributes to controlled output. Yet Wrangler tracks activity by user and project token for that reason. It provides visibility into which models are being used, where they're being used, what they cost, and how their activity mapped into the project. That supports governance today. It may support traceability tomorrow. The original discussions behind this episode emphasize the importance of capturing unexpected learning, creating psychological safety, and connecting discoveries across disciplines and projects. That may be the final lesson from all these stories. We may not recognize the foundational discovery when it first appears. It may look like an odd result, a failed experiment, a short note in Confluence, a forgotten AI conversation, or one sentence that someone almost decided not to share. Our responsibility is not to predict everything it might become, but it is to notice it and to preserve it and to make it safe to share. And lastly, we want to build systems that help it reach the right destination. So we've reached the end of this episode, and I hope it stimulated your thoughts about the importance of encouraging your team members to capture unexpected results within your organization so they're available to them and future people looking for solutions. I've used wikis and products like Confluence and Notion for decades, and I have decades of design history information across all these projects that was effectively crowdsourced by my teams and has served as an important repository of information during both current programs and reference during subsequent programs. I hope this has all been helpful to you, and I invite you to find more podcast episodes, articles, resources, and information about how I help teams work smarter and put AI to practical use at innoguide.net. Until next time, keep learning, keep questioning, and stay curious. I want to thank Steve McIntyre for producing this episode, and I especially want to thank you for joining us. I also invite you to comment on whether you like the format of this podcast. I enjoyed doing it, and I hope you enjoyed it. That's a wrap. See you next time.